Soy director del grupo de investigación llamado Bacteriología, Agrícola y Ambiental, donde nos dedicamos a hacer eso que en el título es suficientemente claro. Nosotros estudiamos bacterias, pero sobre todo en su interacción con suelos y plantas. Y trabajamos tanto con bacterias benéficas como con el control de bacterias fitopatógenas en general. Ese es nuestro interés, el de mi equipo y el mío. Trabajamos para los programas que ofrece nuestro instituto en la Universidad de Antioquia. Para nosotros es un placer presentar al doctor Frederick Cohan, quien es profesor de la Universidad de Wesleyan en Connecticut, en los Estados Unidos. El doctor Cohan, de hecho, ha sido la persona que nos ha guiado en lo que hoy pretendemos nosotros que sea nuestra línea principal de investigación, es decir, es una línea transversal a las demás, y me refiero a la ecología evolutiva. La ecología evolutiva como una forma de entender los microorganismos, cómo ellos funcionan y en general, pues, cómo a partir de su evolución en la teoría evolutiva nosotros podemos entender muchísimos elementos acá. El doctor Cohan es, como les decía, profesor e investigador del Departamento de Biología de la Universidad de Wesleyan. Es ampliamente reconocido a nivel mundial, quizás como la persona que ha hecho los aportes más influyentes en las últimas 2 décadas, pensaría yo, dentro del entendimiento de cómo los microorganismos, especialmente aquellos con mayoría de reproducción asexual, es decir, las bacterias, los virus, evolucionan y cómo ellos se van diversificando. Él nos va a compartir muy generosamente aparte de sus investigaciones y de sus principales conclusiones en 2 modelos fundamentales para él de investigación, que han sido sus estudios en los hot springs de Yellowstone y en Death Valley, también en California en este caso. Bueno, yo pasaré inglés. La dinámica es que vamos a tener una charla por 1 hora, algo más de 1 hora, para posteriormente tener un espacio de preguntas. Quienes quieran hacer preguntas las pueden hacer directamente, entonces tomen atentamente sus apuntes, lo que quieran, y bienvenidos. La idea es que esto sea más que como una simple conferencia, pueda ser como una clase. Pues, y entonces al final vamos a tener espacio para que hagan directamente sus preguntas, o si se sienten inseguros con el inglés, pues las pueden hacer en español. Él tiene un muy buen manejo del español, puede ser que la logre entender, o si no, pues nosotros igual aportaremos en darle claridad ya en inglés. Bueno, voy a pasar el inglés. Dr. Cohan, thanks a lot. I already made a long introduction of you and our group and what this event is. Thanks again for having this time with us. For us, it means a lot, okay? If you want to open your microphone, please go ahead and share your screen. We are here, all yours.
Okay, muchas gracias, Camilo, mi amigo y colegio. Muchas gracias por invitarme. Y es un placer grande visitarlos, aunque sea virtualmente esta vez. Espero poder visitarlos en persona y viajar otra vez a su hermosa ciudad en un futuro cercano. Esperamos. So that's the limit of my Spanish. And the last talk that I gave in Medellin was totally in Spanish, and it was interesting. I mean, I could get all the words I needed. For some reason, in the middle of the talk, I could not think of the word dinosaurio, and I'm not going to use that word today. So, okay, so let me share my screen. Yeah, let's see. Okay. Can you see my cursor?
Yeah.
Okay.
Yes,
great. So nice to be here. So nice to be there, sort of. And yeah, let's get started. So I'd like to start with the big view of biodiversity. We might call it biodisparity, in the sense that when we talk about the diversity of bacteria, we're not just talking about the diversity of this species versus that species, but we're talking about groups that are utterly different. And in our case, they're different in their architecture, genome content, biochemistry, and so on. And one of the things that I'm interested in, and we're going to talk about a little bit, is the origins of phyla. And the one thing I want you to get out of this slide is we take two, this is a phylogeny of phyla, of bacteria. And the thing is that any two phyla, if we go back in their history in the phylogeny, their lineages will coalesce to one lineage that represents one species becoming two species, one species splitting and becoming two species. And that's really what I study, is the processes of lineage splitting, one species becoming two. And there's all this interesting information about how many phyla there are cultivated. It's estimated now there are 1,300 phyla. And I just want to give you a sense of what I think of as some of my favorite very deep groups of microbes. So here we have archaea versus bacteria. And we have here that bacteria and eukarya have a phospholipid bilayer as a cell membrane, whereas the archaea have a single layer, a monolayer. And you know, this is very valuable if you're going to be living way above boiling under pressure, say 125 Celsius. And the thing that I really enjoy about studying the phyla is that the differences between them are not like antibiotic resistance. You know, so we think about what does it take for one kind of adaptation, like an antibiotic resistance, to get from one phylum to another. It's, as we say, a piece of cake, one horizontal transfer and an adaptation that's simple, like resistance, can go from one species to another. But all of these phylum differences are so profound and involve so many genes and so many interactions with the rest of the physiology that they cannot be spread by transfer. And just a couple of my favorites. This one's a favorite, the Firmicutes bacteria, the gram-positives. And they're a favorite because I work with bacillus. And one thing that's interesting about them is that because they have such a strong, thick cell wall of very many layers of peptidoglycan, they're constitutively resistant to osmotic stress. So one of my favorite examples of osmotic stress actually comes from a song by the group America, A Horse with No Name. Some of you might know it. Next time I'll give a Colombian song, but there's a phrase here: the desert had turned to sea. So the idea is you have all these bacteria that are drought resistant. They've been resisting drought and salinity stress for all this time, then all of a sudden it becomes a lake. Well, they don't have to do anything to accommodate because they have this constitutive resistance. And one other example I'll give you is Planctomycetes, a phylum with a stalk. So with this stalk, they can actually hold on to the surface, the sediment, and they can be just a little bit above the surface of the sediment and be getting nutrients that go by. A lot of exciting things. And the thing is, all of these between phylum differences are, they're just not transferable. These represent profound disparities in the microbial world. So what I want to talk about is the process by which the phylum diversification starts, and what it is that we can imagine at some point in the past. Well, we could think of, there's two phyla here today, And if we go back into the past, we'll see they coalesce into one lineage. We might have to go billions of years into the past. But basically, at this time, there were billions of species, microbial species alive, and this was one of them. And this just happened to be one split where the two lineages would persist into the indefinite future. Very few of species could do that. So we have a huge amount of time, and these happen to be two lucky species. So I think of the rate of phylum origination as being the rate of speciation, which is happening quite frequently, and I'll discuss that, times the probability of persistence of any one lineage, which of course is near zero into the indefinite future. So I'm not going to talk so much today about the persistence into the future, although that's something that I think I discussed 2 seminars ago in Colombia at a Latin American microbiology meeting in Cartagena, which was wonderful. Okay, so when I talk about species and the origin of species, I'm not talking about Escherichia coli or Bacillus subtilis. The named species of bacterial taxonomy are extremely broadly defined. And there is a lot of ecological diversity within a bacterial species taxon. So I want to talk about a much more limited, smaller group of bacteria that I call ecotypes. And I want to argue that these ecotypes have or can have the properties that we like to attribute to species in biological taxonomy. So there are two properties of ecotypes I want to insist on. One is, if you have two ecotypes, they have to be ecologically distinct. And also, within an ecotype, we expect that there will be ecological homogeneity. So that is, we have two ecotypes, each one consists of interchangeable bacteria, but the two ecotypes are different. So here I want to show how this works. So let's say we have ecotype 1, this is the greenish ecotype, and here we have a purplish ecotype 2. And so we can say, okay, these two ecotypes will say they're ecologically distinct from each other. The tricky part is, are they each homogeneous? Well, I mean, anybody who knows something about bacteria knows that you cannot expect there to be absolute equality of ecology among close relatives. And so I allow for that in my ecotype definition. So the deal is that we allow within an ecotype, there can be various ecologically distinct lineages. So in this case, we have kind of a lime green lineage, and here we have a forest green lineage, and here we have a Kelly green lineage. And I don't know how well those green shades translate into Spanish, but you can appreciate they're different. And what I want to suggest here is that these three different ecologically distinct lineages are not ecotypes because they're not different enough to coexist indefinitely into the future. And so it turns out that over time, we lose this lineage, because they're just too similar to the Kelly green ecotype, the Kelly green lineage here. And likewise, we have this purple and blue ecotype here. Well, the purple and blue are doing ecologically different things, but the blue one is not different enough that it can coexist indefinitely with the purple one. So what I want to talk about today is how these splits happen, like how we can go from one ecotype in the past to split to become ecotype 1 and ecotype 2, and what allows the lineages to persist into the remote future. So just going from to more general ideas of ecology, of botany and zoology. So what are the ways that ecotypes could coexist out of ecological differences? Well, let's start on the right side here. One possibility is that different, and these are different bird species here, that these bird species can coexist in the same place because they use different resources in the same habitat. So this crossbill bird eats this species of cone, and this one eats this species of cone, and so on. So they're diversifying by the resources they use in the same place. But then we can also have something that was invented by botanists back in the 1940s, this idea that within what we call a species, in this case, a species of Achillea or yarrow, a plant species, that here there are multiple ecologically distinct populations, and each one is superior at a different elevation in California. And this is something that I actually studied a bit when I was in college. So I think that's interesting. So we have two different ways that you can be ecologically distinct to provide a kind of coexistence into the future. And now I'd like to give a more microbiological view of coexistence by what we call niche partitioning, or just dividing up the resources into different parts. So one group of studies I'm going to talk about is our Yellowstone study done in collaboration with David Ward at Montana State University. And then there's the Bacillus study that's been led by my group at Wesleyan. So here we have differentiation by chemical or physical conditions. So here we have a hot spring that's bubbling out of what I call the bowels of the earth. And the water dribbles slowly down the hill. And we have a temperature gradient starting at 68 here, going down to 65, then 61, then 57. And there are different ecotypes, very closely related ecotypes of Synechococcus that are adapted to different temperatures, different physical conditions. Likewise, in Bacillus, in this particular case, we can have different ecotypes of Bacillus that are very closely related that are adapted to different levels of solar exposure. So the idea, so this is a west running canyon where there's this arroyo in the middle, so the canyon is running this way, and we have the south-facing slope here, which is high. Is that a question? Sorry. Okay, so we have the south-facing slope here, which is hotter, and the north-facing slope here, which is cooler. And then it turns out we have ecotypes, and you'll see in a moment that we have ecotypes that are adapted to the north face, south-facing, and the north-facing slope, and also the arroyo for that matter. Okay, and then we can also have in the microbial bacterial world, we can have differentiation by resources in the same microhabitat. So this is one of my favorite sets of studies by Richard Lenski and Zachary Blount in E. coli. This is their experimental evolution study, which has gone on for something like, I don't know, 40 plus years now. And what they found is that, well, it's not really sugar A. We have different organic compounds that the ecotypes in this long-running experiment are adapted to. So we have one ecotype is adapted to glucose, and another one is adapted to citrate, which is not a sugar per se, but it's another organic compound. Then in the bacterial world and the viral world also, which I'll be talking about soon, we can have differentiation of pathogens by tissues or host species. So in the case of Streptococcus pyogenes, we can have strep throat ecotype. Within this species, Streptococcus pyogenes, there are multiple ecotypes, and one is a throat adapted, causing strep throat, and one is a skin adapted, causing impetigo. So those kind of makes more real the ways that very close relatives in the bacterial world can divide up the habitat, divide up the resources. So let's talk about whether these ecotypes have species-like properties. Excuse me, I just want to get some water. And bienvenidos a nuestra cocina. So, so what are the species-like attributes that ecotypes might have? Well, the possibilities are, and I'm just going to say these are two ecotypes here. The possibilities are, one, they are, well, we know we're defining them to be ecologically distinct, so check. They're cohesive in the sense that the diversity within each ecotype is constrained in some sense, irreversibly separate, that ecotypes can just diverge and diverge and diverge, and that they will form separate DNA sequence clusters. And so I'm going to show you how this works in a particular model that we developed that we call the stable ecotype model. So imagine here a world where we just have one ecotype, this purple ecotype, and these circles represent the different genomes of members of ecotype 1. And then we postulate that there's an adaptive mutation in this one genome here that allows this cell to utilize a new ecological niche, a new resource, so that in the next frame, we see that that purple genome is disappeared here, but now it appears as a member of this new ecotype. It is the founding member of this new ecotype. So that's one part of our model, that we can get origination of new ecotypes by mutations, by horizontal transfers, by various mechanisms to become ecologically different enough so we can use new resources. Okay, so that's one thing. Then now let's suppose that we have two coexisting ecotypes. And I want to make here, first of all, the case that each one of these is cohesive in a sense. So here is the idea that we have this diversity within ecotype 1. They're all using more or less the same resources. And let's say this was a mutant here, this triangle. It's a mutation that allows this cell to be superior to everyone else in this ecotype. So this cell now outcompetes everybody else. And because recombination is rare in bacteria, what's going to happen is that this entire genome becomes all that's left in the future of this ecotype. So there's this purging of diversity, this loss of diversity. And so this speaks to the theme of cohesion within an ecotype, because owing to the rare nature of recombination events, any adaptive mutant can proceed to become 100%. This whole genome becomes all there is of diversity within this ecotype. And then I also, and these events, these selection events, which involve a purging, a whole genome purging of diversity, we call these periodic selection events. Now, I also want to make the case here that these two ecotypes are irreversibly separate in a sense, because of their ecological distinctness. When this population has a selection event, it might purge the diversity within that ecotype, but it does nothing to the diversity in the other ecotype. So ecotype 2 persists into the future. So we have a kind of a long-term coexistence that's possible for ecologically distinct populations. Now, so we have a kind of a long-term coexistence of ecotypes, but what do we mean by indefinite coexistence? Like, this is a picture, it's a rare photograph of the day the comet fell 65 million years ago. And we're lucky to have this photograph, I would say. But anyway, what we have here is that we can't expect that random lineages that we're studying are going to persist past some really catastrophic event like that. So what do we mean by indefinite coexistence in the sense of the origin of species? So I want to give you a much more limited notion of indefinite coexistence. We'll just deal with one particular aspect of it. So I'm going to start on the right again. So this is the stable ecotype model, where we have one lineage, the blue lineage, it splits and a red lineage is founded. And then what I want to say here is that, oh, so these letters represent the resources that each ecotype uses. So this one uses resources A, B, and C, and this uses A, B, and D. Now notice that the red ecotype uses a resource that is not claimed, it's not used by the blue ecotype. So what that means is that the blue ecotype can have lots and lots of periodic selection events, and yet it will never extinguish this other ecotype because this one always has D. I'm assuming these are interchangeable resources. So this guy has a resource it can use that these guys cannot use. Oh, by the way, I should explain this figure and what it means. So we start with the blue ecotype, and what I'm indicating here in the dashed lines are diversification that occurs within the ecotype. So just by random mutations, we have a growth from this point here to a diversity of sequences within this ecotype. But then we have this adaptive mutation events. And I realized to make it consistent with the previous slide, it should probably be a triangle, not an asterisk, but I'll fix that for the next seminar. So when we get this periodic selection event, it means that the rest of the diversity is purged and only one lineage goes forward. And then after this periodic selection event, again, we have diversification within this lineage, and then followed by a sweep, a sweep of diversity. And what I want you to notice, and I think I've got some better slides of this, but what you see here is that after there are two ecotypes, this lineage has its own periodic selection events, and when they occur, it purges the diversity within the blue lineage, but it has no effect on the red lineage. And likewise, when the red lineage has purgings of diversity, it doesn't affect the blue lineage. So what I want to say is special about this particular model is that there is no mutation in the red that can extinguish the blue because they each have unique resources that the other one is not using. Okay. So let's contrast that with what I call the nano niche model, which is, which I'll say is kind of a stupid name. I thought of it. There's probably something better, but we're stuck with it. So here's the idea that within an ecotype, well, so there are various ecotypes at any one time, and there are periodic selection events within this green ecotype and within the red ecotype, but at some point, this blue ecotype outcompetes, extinguishes, brings to extinction the green and red ecotypes. Well, how is that possible? Well, what's different between these two models is that there are no unshared resources here. All these resources are shared. They all use A, B, and C. But in this case, the magenta ecotype uses mostly A and a little bit of B and C. And here, the blue ecotype uses mostly B, a little bit of A and C, and so on. So these ecotypes are only quantitatively different in their resource differences. So what that means is that there could be a super adaptive mutation in the blue lineage here that actually will outcompete to extinction the magenta and the red. In fact, we've seen this in the past, that here we have the blue ecotype and it formed this, I guess that's brown and yellow, and then we have little periodic selection events within the blue, but then we have one of these super adaptive mutations where the blue ecotype outcompetes to extinction the brown and yellow So that is, so what I want to say about indefinite coexistence is that I would say that that a system like this of 3 ecotypes, that I would not call these indefinitely coexisting, because since they're not using different resources, they one sister ecotype can outcompete another sister ecotype. Whereas in this case, where there are unique resources used, then it becomes harder for one ecotype to outcompete the other. Okay, so that's the idea of this indefinite coexistence, a kind of an indefinite coexistence that doesn't go beyond global catastrophes, but at least guarantees that one sister ecotype will not outcompete the other sister, that they're different enough so that will not happen. Then also, I want to make the case that the divergence between ecotypes is genetically irreversible. So here, so think about this notion that we have in zoology and botany. So the idea is that if we have 2 very new species of animals, the idea is they have to restrict their genetic recombination between each other, or else the species will kind of fuse and become one population. So what I want to argue is that fusion of ecotypes can never happen as a result of recombination, recurrent recombination between populations. And I think, let me see how I'm doing for time. Yeah, I'm going kind of slow, so I think I won't actually explain it. I'll just assert it, that recombination bacteria is so rare that when there is recurrent recombination between ecotypes, it cannot reverse the ecological divergence between them. And I like to say that sexual isolation is not an important milestone for speciation. It's just not needed. If 2 lineages are going to become more and more and more different, they do not need to evolve sexual isolation, or that is resistance to recombination with each other. And so this is what we see, and this is an experiment from my lab, that if you put 1 clone of bacteria in a vessel and let it evolve, you can get what we call sympatric speciation. That is ecological diversification in the same place. And I just want to point out what is different about these 2 populations, that this one lives as a raft. It floats on the surface of the water, and this one lives in the water column. And they both evolve in the same vessel. And how is that possible? Well, there was not enough recombination between them to stifle, to hinder, to prohibit the divergence between them. They could just diverge and diverge and diverge. And this is what we believe is happening in the bacterial world all the time. So with that as a background, I want to talk a little bit about how we're using our theories to discover and characterize the most newly divergent bacteria. And before I get started, are there any questions that I could just slow down and answer the questions you might have? And I just might mention, me siento que he perdido la audición, y Camilo me ayudará. So, question, preguntas? Okay.
There is one in the chat. There is one in the chat, Professor.
Okay, go ahead. Can you, let me see. Yeah, okay, hold on. Chat. Okay, so the one from Christian Serna. The ecotypes can be understood in a context of responses for pre-adaptive mutations. Okay, now hold on. Share. Okay, so I guess not the other thing. So the ecotypes can be understood in a context of responses to pre-adaptive mutations. Well, I understand the ecotypes as being, I mean, it's possible I'm not quite sure I understand what you mean, I'm sorry to say, but I think that an ecotype could be founded by a single mutation or a single horizontal transfer that will allow a lineage to utilize some new ecological niche. And as time goes on, the ecotypes can become, I'll say, more entrenched, can be more highly and specifically adapted to their habitats and their new resources. But I'm not sure what you mean by the pre-adaptive mutations. And if you want to add to your question, I'd be happy to address it further.
Cristian si está escuchándonos, no sé si quiere. Okay. Here is his comment. He's saying that you got the question right.
Okay. Yay. Okay. Any other questions while we're on stop screen?
I do have one.
Go ahead.
If Professor Camilo allows me to.
Claro, Sebastián, dale.
Saludos de Alemania, por cierto.
Hola, hermano, dale. This question is coming from Germany, Dr. Cohan. Sebastian is a former student in our department, but he's now working in Germany. Okay, go ahead, Sebastian.
Thank you so much, Professor. Would your ecotype model and your niche model be could it answer for the evolution of stromatolites in like the fossils of cyanobacteria with other types of perhaps some other types of bacteria? Because there's this question that stromatolites are sometimes the same niche, and sometimes it depends on different factors like the growth of the type of cyanobacteria or the salt water that got to it, whatever. So we are not sure, in studying astrobiology, we are not really sure if we should what type of evolutionary theory or hypothesis should we take into account on studying them?
Okay, thank you. Well, so the way I like to think about it is, let's suppose that, and you'll see some phylogenetic trees that I'll be showing, but whenever we have, I'll just use my fingers here, whenever we have one lineage that's become two, and they coexist and persist for quite some time. So we might have two ecotypes that have been around for, let's say, in the millions of, in a small number of millions of years. So the way I see it is that in order to coexist for a very long time, they at any one, so we're talking about different global changes in the meantime, perhaps different communities that they might be living in, and so on, that I think for two lineages to coexist, and let's say they're going into the future as the communities they live in are changing, it has to be the case that at any one time, that at every single time point, there has to be an ecological difference between the two over the long haul of time. And if it ever happens that two lineages that are at one point in time, they are ecologically distinct, but then as communities change, at one point, if they are no longer different from each other, I think we can say that they will not coexist, at least in the same place, for the indefinite future. That long-term coexistence is probably going to require a niche diversification of some kind. And the point I'm trying to make is that the niche differences have to exist at every time point along the line. That if ever there were a time in the past that two lineages became ecologically similar owing to changes in their environment and who they're interacting with, then that would be a dangerous time for their coexistence, and their coexistence might not persist indefinitely at that point. So I don't know. I mean, what I'm saying applies, I think, beyond stromatolites, but I think it's interesting, the point that you make, that I guess, you know, and I don't know enough about stromatolites, but that maybe there's evidence that the different populations we see probably weren't ecologically different, but, you know, how could we know looking back in the past that far?
Still, your discussion is amazing, and I will surely read your work. Thank you so much, Professor.
Oh, you're welcome, and I'll be happy to give. Well, in fact, if you look at my title slide, and I'll make my slideshow available to you. I'll send it to Camilo. But so anyway, the title slide gives my publications, and I'd be so delighted to have you read them. So let's see here. Okay. Yeah, okay. So yeah, so at this point we have a model for what ecotypes are, and I want to use that model and the way of thinking about things to try to figure out what is the genetic basis of ecotype origination, and what are the ecological dimensions along which ecotypes diverge, and are there species-like characteristics held by bacterial ecotypes. And this part is something, this last part, the tempo of bacterial speciation and how it's affected by lifestyle. And I think this is the most speculative part of the talk, but I think it's pretty interesting. Let's see here. Yeah, okay. So let's move on to the bacillus part of this talk. So I've already introduced this system to you. This is this radio facility WASH in Death Valley National Park. And we have these two environmental contrasts. We have the south-facing and north-facing slope. We also have a silty soil and a gravelly soil, and that'll become a little more important in the future. So what we've been doing is we isolate, you know, we've isolated many hundreds of strains related to Bacillus subtilis from Death Valley National Park. And this is an early phylogeny based on 3 genes. And so here's where it's rooted to the rest of the world. And what I want to make the point is that we can look at a phylogeny and detect the ecotypes just by looking at the phylogeny without knowing anything about ecology whatsoever. So the idea is that, again, we have this blue ecotype and red ecotype. So, and here we're presuming that there are multiple periodic selection events, like multiple sweeps, loss of diversity within each ecotype, and then the ecotypes are split, and then we continue to have these losses of diversity within each ecotype. So let's just suppose that we were sampling, we didn't know there were ecotypes, we're just sampling close relatives, and then we find that there's a cluster here and a cluster here, just based on sequence divergence. And when we see that, we'll say, oh, that is consistent with these being 2 ecotypes, because the amount of diversity we see within one ecotype is only the diversity that has accumulated since the last periodic selection event, since the ultimate periodic selection event. So we have just a little bit of diversity that's occurred within this one since this last event, and then a little bit of diversity here. So whenever we see among close relatives that there's a cluster of close sequences here and a cluster of close sequences here, we can say, oh, these are likely to be ecotypes. So then we look at a real tree and we say, oh, well, maybe this cluster here is an ecotype. But you know, it's trickier than that, because when we look at something like this, is this a cluster that equals an ecotype? Or is this larger cluster equal to an ecotype. So we developed this algorithm we call ecotype simulation that I won't really explain, but what it does is it looks at a whole tree and estimates the rate of periodic selection, the rate of speciation, that is the rate of formation of new ecotypes, and then it demarcates the sequences into different ecotypes. And that's just what it does. We've been using this. And so the thing is, whenever we do this kind of thing, we find that so PE means putative ecotype. And so these are ecotypes that we're hypothesizing as putative ecotypes. And it turns out whenever we do this, we always find that the ecotypes are ecologically distinct from each other based on where they're found. So for example, PE15 is found mostly, overwhelmingly, 73%, I don't know if you can read that, but 73% in the north-facing slope, the NFS. So this is the cool-loving ecotype, whereas other closely related ecotypes are, this one is found only, this PE21 is only found 38% in the north-facing slope. And if I could read this slide at the top, because of all this chazarai at the top here, I can't see it, but there are lower levels in the north-facing slope. So we always find that the ecotypes that we hypothesize are ecologically distinct. But what we'd really like to know, and it's more difficult, is whether the ecotypes are each ecologically homogeneous. That is, do we have enough resolution from just 3 genes to decide that this ecotype, this PE15, are they really ecologically and genetically the same as each other? Well, I want to introduce what I call the Doolittle model of very little species. This is Ford Doolittle, who he and I have discussed speciation for decades. And his idea, which I agree with in some cases, is that speciation is happening every day. That with horizontal transfer happening all the time, that the only bacteria that aren't exactly the same are going to be a mother and a daughter. And you're going to see to some extent that seems to be true in bacillus, in the sense that we're going to find that these putative ecotypes of bacillus are not really homogeneous. And we're going to use some genome technology to show that. So this is a preliminary bit of work, which we actually published in 2014. And so what we're going to find is that bacillus speciation is really very rapid. And what I want to say is, so we sequenced 5 members of, well, we sequenced 4 members of PE15, and we had one freebie that somebody else had sequenced. And what we find here is that every one of these 5 strains in PE15 are distinct in their genome content. But more interestingly, in a way, and I'll explain in a moment, that what we've done is we've looked at the core genome, that is the shared genome, the genes that are shared across the PE15 strains, and asked whether there are different histories of natural selection on the different lineages. And it turns out that every one of these 5 lineages, these 5 strains, had its own unique history of natural selection. That is, this one didn't have any positive selection, but this one had 2 genes that where natural selection had acted on them, and those 2 genes were not seen in the responses of these 2. So basically, the genes that are changing, that have changed over each lineage, are a unique set for each one. And I just want to shout out an idea by my colleague Michiel Vos, who has hypothesized that when we find that closely related strains are different in their selection regimes, in their histories, he says that is evidence that they're actually ecologically distinct. Why else would they have different histories of natural selection? Well, I think there are reasons we could imagine, but anyway, so what he's arguing is when we see this is that what we thought was one ecotype based on our phylogeny is actually 5 different ecotypes within the putative ecotype 15. So there's a lot of diversification, very rapidly going on in bacillus. And I want to mention, so I want to look at, so what kind of diversification is going on? What is the nature of the genetic and ecological differences between these ecotypes within PE15? And I'm going to argue that they kind of subscribe to the nano-niche model. That as we look at different members of PE15, it's like they're only, I'm going to argue that they're only quantitatively different, where they use the same set of resources, but each strain focuses on a different subset of resources. So now the thing is, most bacteriologists will say that this is kind of a bizarre model of species, because in bacteriology, we like to think of yes, no, present, absence of metabolic features, that you can do this or you cannot. We tend not to think of, well, you use this one a little bit better and use the same thing a little bit worse in some other strain. But I want to make the point that this quantitative diversification happens all the time in animals, and why not also in bacteria? So I'll give an example. This is something I talked about earlier. So these are 3 species of crossbills from the same forest in England. And we say that, yeah, the parrot crossbill eats the Scotch pine, and the common crossbill likes the spruce, and so on. But here's the thing. They are not, they are only quantitatively different. That is, the parrot crossbill prefers the Scotch pine, but it will also eat spruce and larch, and vice versa. So this is really like the nano-niche thing. These guys could last for a long time coexisting because of their ecological differences, even though they're really sharing resources. So I want to give you the evidence that the PE15 genomes that we've looked at are actually only quantitatively different in their ecology. So what we did is we looked at all of the gene content differences among the 5 strains, and we focused on two functional subsystems that related to ecology. So we're not talking about DNA repair, gene content differences. We're talking about ones that deal with taking up resources from the environment and utilizing them. So it turns out there are only 2 functional subsystems that differed between our 5 strains. That there's this one strain, this G1A4 strain, that had extra genes for maltose and maltodextrin. So it had 9 unique genes, and 3 of them were paralogous to genes that other strains had, and 6 were just entirely new maltose, maltodextrin genes. And for inositol, there were 3 extra copies, 3 paralogous genes. So it turns out, so here's the thing, that everybody, all of the PE15 strains, they all can use maltose and maltodextrin, and they can all use inositol. But what makes this strain, this G1A4, unique is it has extra genes. In fact, it has a unique pathway to using maltose and maltodextrin, but they can all use maltose and maltodextrin. So here I'll show you an experiment that we did. So this is growth with only maltose as the only carbon source. And you could see over time that the other strains from the desert that we tested, they can use maltose and they grew. But the strain with the extra maltose genes, it grew the most. So this is kind of a quantitative speciation, that these guys, probably by virtue of having 6 extra, well, actually having 9 extra genes for maltose utilization, they can use a resource that everybody else can use, but they use it better. And so it's kind of a quantitative speciation that we're looking at here. So what I'm arguing here is that Bacillus seems to be rapidly speciating. And I want to, and we could expect that Bacillus would be a rapid speciator because it's a generalist heterotroph. That is, it's just using carbon sources that are out there. So all a generalist heterotroph needs to do to be able to use a new carbon source is to grab by horizontal transfer a new pathway. It's that simple. And we can expect that there will be lots of speciation diversification in the Bacillus and more generally the generalist heterotroph world. So what I'd like to move on to now is a phylum that is a phototroph, photoautotroph phylum, the cyanobacteria, where I want to argue that speciation is expected to occur much more slowly. Now, before I get into that, I'm wondering if this would be a good point for questions that you might have about the Bacillus system and maybe more about the theory that I've been talking about. Because I'm always looking forward to an opportunity to slow down. So go ahead and be my guest if you'd like to ask a question. Okay, well, I'll move on to Synechococcus, hot spring Synechococcus.
Professor, there was a question.
Oh, please, go ahead.
No, in the chat.
Oh, okay. Okay, hold on. Okay. Okay, yeah, from Juan Camilo Arboleda. What do you think about the species concept in bacteria? Is it useful to base this concept on nucleotide identity, or should it be based on a more ecological ground? So Juan Camilo, I love that question. And this is something I think about and don't really have a complete answer to. But I'll be addressing it a little bit more as the talk goes on. But let me answer your question right now. That in terms of trying to understand the origins of diversification, I think we have to look at the ecotypes. That is how one ecotype becomes two ecotypes, recognizing that there could be hundreds or thousands of ecotypes within a recognized species taxon. That is the species taxa of bacterial systematics. Now, so I think when we're talking about the dynamics, the population dynamics, we really do need to talk about an ecologically based definition where each ecotype is homogeneous, and the different ecotypes are ecologically distinct. But what I'm going to be talking about toward the end of the talk is in what sense the species taxa have some biological reality. And I will say they don't deserve to have biological reality. The people who invented and demarcated these species taxa in the middle of the 20th century, they were so lucky to have come up with these species demarcations that actually have a biological basis, because those folks were not interested in a species with biological basis, but I am. And you'll see in a moment that we're going to talk about a molecular basis of species taxonomy. So I hope you'll look forward to that. And that's as far as I'll get now. And so Dr. Cohan, this is Christian Serna. Maybe it's a mix of both concepts, the molecular and the ecological. Well, it depends what you want to do. If you're studying population dynamics and evolutionary biology, it's the ecological. If you're talking about how to characterize the larger diversity and put it into a taxonomy, I think the molecular, the broader molecular species will be more useful. And we'll hear more about that later. And Juan Camilo says, thank you very much. And I'll say my favorite phrase from Colombia, which is con gusto. That is so much better than de nada, don't you think? I mean, con gusto, with pleasure. nice. It's wonderful. Okay. So let's see. So let's go back. So yeah. So let's go to what I'm going to call a more slowly diversifying phylum, the cyanobacteria. So we're not, in this case, looking at just any old cyanobacteria. These are hot spring cyanobacteria. And one of the reasons that I suspected that speciation would be slower in the cyanobacteria in general would be that they have lower metabolic plasticity. So they don't have habitat partitioning by organic resources. So whereas bacillus can just speciate, take up one new carbon, another carbon, these guys don't use carbon. So it really, I would think, slows down their speciation. And also, the limit of habitat partitioning is by, in this case, by their horizontal and vertical dimensions. The horizontal dimension being by the quality and quantity of light that makes it through the mat. That's vertical. Yeah, the vertical is the quality and quantity of the photons, and the horizontal is both the temperature that's falling as you go down, down the mat, down the spring, and also the mineral resources decrease also. Whereas these guys at the top of the spring, we found by genomics, didn't expect it, but we found it. These guys, the ecotypes up here, they can use phosphate as a resource because they are the first ones, the first bacteria to absorb and to consume the phosphate. But down here in the cooler reaches, there isn't any phosphate left. So we find by genomics that these ecotypes focus on phosphonate, a more difficult to absorb phosphorus ion. So things like that are very interesting. Now, on top of just the fact that this is a photoautotroph, also just that it's a pretty limited community and there's just less opportunity for horizontal genetic transfer because there are, so I want to slow down, there are just so few species in a spring like this, so we'd expect that there's less opportunity for horizontal transfer and thereby perhaps less speciation. So once again, we took these phylogenies. These are just two different clades. We analyzed them separately. The hot clade, the A and A prime-like clade, and the B prime clade, a cooler clade. And we ran our ecotype simulation, and we found all these putative ecotypes. And so the first question we ask, are the hypothesized ecotypes ecologically distinct from one another? And we'll say, yeah, that's the thing that we always find out. So in this case, we took different ecotypes and took the ecotypes that were known to be at different depths of the mat. And it turns out that that ecotype, the B'9 ecotype, that was found higher up in the mat, that it has an optimal growth at this, this is, what is it? Yeah, this is the amount, these are experiments, this is the amount of light coming in. And so the B'9 actually does best high up with lots of photons, and A1 is adapted to a lower level of photons, A4 lower yet, and A14 very low. So we can see that the habitats where they're found in are actually where they grow the best. And so that's the ecological distinctness that, yeah, they are, well, number one, you could just say they're ecologically distinct based on replicably the ecotypes always being found in different habitats, but we find out that there's a physiological basis for their habitat differences. And so is there, and there's actually a bunch of slides I could show for this, and I'm just going to show one which may or may not be the best one. So to what extent can we say that within an ecotype, a putative ecotype, that there is homogeneity among all of its members in its ecology? So bear with me in this one, it's a little complicated. So this top panel here, represents putative ecotype A1. And these different colors represent different high frequency sequences. So there were 4 high frequency sequences. One, the blue one, which is always very high frequency. And these guys, they're fairly low frequency, but the thing is there are tens of thousands of members sampled here. So now, what is the x-axis? The x-axis represents different habitats that we sample. So they could be different springs, they could be different temperatures in the same spring, they could be different depths in the same spring, they could even be different counties in Wyoming. It could be all sorts of things. And so what we see here is we predicted that if all the different sequences that are within A1, if they're all ecologically interchangeable, then we should see that as we look at these different sequences over different kinds of habitats, there should be basically each sequence should keep its same relative frequency. So the blue sequence is always the highest, and these other ones are always very low. So what if it happened that the red sequence was actually adapted to one spring and the blue guys were adapted to another spring? Then as we look across different springs, we would see the blue going down and the red going up. But the fact that blue is always high and these red, green, and purple are always low indicates that they're interchangeable. Their relative fitnesses stay the same no matter what habitat you look at. So that's kind of an indirect evidence of homogeneity within this putative ecotype. And this is basically what we found in every putative ecotype. Here is A4. Here we just have two high frequency sequences, the blue one and the red one. And we see there's a lot of commotion going on here. And there are actually fewer habitats where they were sampled compared to A1. But basically you see that the blue one is always in the higher frequency, the red one's always in the lower frequency. And we see that again here in A14, where there are 3 types: the blue sequence, the red sequence, and the green sequence. So that is one kind of evidence we have that there is homogeneity within what we're calling one putative ecotype. And then we did some genomic sequencing of these ecotypes, and we found that within an ecotype, there was a 0.5% genomic divergence. That is what we call the ANI, the average nucleotide identity, was actually quite high, a 0.5% divergence. So this is saying that in the cyanobacterial world, there can be divergence, there can be divergence and divergence up to a 0.5% genome difference, and yet we don't see a speciation event. Speciation is happening quite slowly in the cyanobacterial world. Now, so that's all I'm going to say about our Synechococcus system. And now I want to move to a really cool metagenomic study that's not actually by me or my collaborators, but I did publish a commentary on it, which I think is pretty interesting. And I'll basically explain to you what my commentary says. So this is what these folks did. They did a metagenomic survey of this acidic lake called, I can't remember if it's Minnesota or Wisconsin, but it's in the upper Midwest of the US. And it's an acidic lake called Trout Bog Lake. It's a bog, basically. And what they did is they assembled genomes from the metagenome sequence. And they sequenced up to 2% divergence. And they gave their clusters, what they called a genome, they gave them names. So this is Chlorobium 1280. That is, it is a box of sequence that's all very similar, and it's in the Chlorobium genus. And they have Methylotenera here, and just gonna try to make this a little bit easier for me. Yeah, okay. So basically what's happening here is they looked over time, and the paper they ended up publishing goes longer in time, but this is basically it, that they're looking at any spot in the genome. They're asking how much diversity is there? And when a spot in the genome, which is represented by this y-axis here, when there's a spot in the genome that's 100%, it's like dark blue. So what they found out over the time of their study, over time, is that there was a full genome sweep of diversity. The whole genome is swept. I call this a garage broom sweep, right? And you'll see the alternative being a whisk broom. And I couldn't actually get good translations for these words, but you guys recognize the picture of a garage broom. And so what we see here is this huge, up to 2% diversity within this box, that it gets totally swept across the genome. And similar things are happening, well, to some extent in the Methylotenera. Now, what is interesting ecologically about Chlorobium and Methylotenera is they are both groups that I would predict would have slow speciation. The Chlorobium, because it's a phototroph, and phototrophs, it's like the Senecacoccus thing. How much opportunity for diversification is there in a phototroph? Like, they don't use carbon sources. So what we see here is that apparently the whole 2% of divergence here can get swept by one periodic selection event, a whole garage broom sweep. And we see something similar happening here in this Methylotenera group here. And Methylotenera is similar to the phototrophs in that there's not a lot of what I'll call metabolic plasticity. That is, Methylotenera bacteria use only one carbon source. They use the C1 carbon source methylamine, and that's all they use. So there's no opportunity for speciation based on using a different carbon source. So I would predict that speciation is going to be slow in most C1 organisms and most phototrophs. And what we see here is that in the context of 2% genomic divergence, we see there is pretty much an entire sweep of genetic diversity, a garage broom sweep. Now let's look at other things that can happen. So we can also have what I call whisk broom sweeps. So this is the case, so this is from the Bendel paper. This is Actinobacterium, which is a generalist heterotroph, kind of like Bacillus, maybe more so. And what we see here is that there are a lot of blue spots, meaning that there is no diversity here and here and here and here, but there is diversity elsewhere. So what we see is that I'm going to call these whisk broom sweeps, that the diversity just at this one place is kind of swept by a little whisk broom. And what I want to argue is when you see whisk broom sweeps in some taxa, it means that that speciation has been rapid. And I'll explain this a little more carefully. So this is from my commentary on the Bendel paper. So let's say that we have one group here, like the Chlorobium, where there is a whole genome sweep. So here what we're going to say is within the whole 2% of genetic diversity, of genomic diversity, we say, yeah, these different dashed lines represent different sequence variants. But when we get an adaptive mutation in this genome, what happens is that whole genome outcompetes everybody, and now we get the garage broom sweep. Now, how should we understand a whisk broom sweep? Well, I'm going to skip to the bottom panel here. And the hypothesis to explain a whisk broom sweep is that let's talk about, let's say, a cluster within Actinobacterium. And let's say it's the 2152 cluster, named cluster of Actinobacterium. So let's say that within a 2% genome sequence group, that there are actually 3 different ecotypes that we don't know they're there, but they're there. And so let's say there's a blue one, a green one, and a red one. So let's say, first of all, that there's a periodic selection event within this blue ecotype. And what happens then is that there will be a whole genome sweep, a garage broom sweep of diversity here because it's just one ecotype. Now, here's the tricky thing. What I'm going to suggest is that this adaptive mutation here might be generally useful so it can pass from the green ecotype, from the blue ecotype to the green ecotype. So now we have this homogenized blue ecotype, and the adaptive mutation moves by horizontal transfer into the green genome here. And now this green individual is better than the other members of its ecotype. And so the next frame is that they all become the same. But notice that what has become the same here is a mostly green ecotype with a little stretch of blue that represents the adaptive mutation that moved from the blue. And then you see the same thing in the next frame, that the adaptive mutation in the green moves over to the red ecotype and causes a periodic selection there. Now, I want you to notice here, what is it that is the same across these? There's just this one little bit of blueness that is homogenized across. So I'm suggesting that this is how we get a whisk broom sweep, that it requires that a block of strains, a cluster that's in this case is within 2% of sequence divergence, that there must be ecologically distinct populations within it where an adaptive mutation can pass from one population to another, yielding this one little bit of the genome that gets homogenized. And that gives us a whisk broom sweep across that adaptive mutation. And what I want to argue is that when we look at the garage broom sweeps, we tend to see that these are groups that would be expected to have slow speciation. So photoautotrophs, we also saw Bendel et al. Showed a garage broom sweep in Gallionella, which is a chemolithotroph. Again, not much room for ecological diversification. And we have Rickettsia, an obligate intracellular parasite, which probably doesn't get around that much, and so on. So then we have the whisk broom sweeps, which are groups that we would expect a priori to have rapid speciation. And so I'll just talk about this one here. This Acanthopluribacter is a generalist heterotroph. So we should expect that it would have rapid speciation. And indeed, correlated with the rapid speciation that we expect, we see that it has whisk broom sweeps. Okay, so let's see. I want to make sure we have time for questions, so I might just skip some stuff here. And yeah, I just want to talk a bit, just because I know this is of interest to Camilo and David. What do we know about the diversification within Bacillus, that is Bacillus subtilis, among the most closely related ecotypes? And one of the things we found is that in that the Connor paper, that we found that there were some ecotypes that tended to be found on the south-facing slope, like the PE12, and there are others that tended to be found on the north-facing slope. And it turned out that there was a correlation between the slope that an ecotype was found on and what I call the heat adaptation index of its fatty acids. So in other words, there are some warm-loving fatty acids that tend to be rigid, and they tend to be found in like the PE12, whereas the cool-loving fatty acids that are more flexible tend to be found on the north-facing slope. And this is work by Nora Connor, my student, and my collaborator, Johannes Sikorsky. And then recently, my master's student, Nicole Delgadio, was looking at to what extent there's ecological diversification in bacillus with regard to elevation and rhizosphere associations. And rhizosphere associations, I know, are always of interest to my colleagues at Antioquia. So let's look at what Nicole and her team found. So, so one thing we found, this is looking at the Bacillus atrophaeus clade, which is very closely related to Bacillus subtilis. And what we found is that there was only rare diversification by elevation, that there's just this one ecotype here that tended to be found at low elevation, but everybody else was found at high elevation. So just not much going on in terms of elevation adaptation. But there was a lot of evolution with respect to rhizosphere associations. So the green means rhizosphere associated, and the brown means free soil. So you can see there were some ecotypes. Here we have 16 isolates, where 3/4 of them were free soil, but we have other ecotypes that tend to be mostly rhizosphere. So we thought that was pretty interesting, and we'd really like to know something about why that is and what the genetic basis of that might be. Then I just want to show you by elevation, if we look at Bacillus, this is the elevation here, that Bacillus atrophaeus is just kind of bizarre, that it just really loves the high elevations. This is 3,000 meters, and there was only one ecotype that was totally found at lower elevations, whereas the other species, and these are now updated. This is now Bacillus species-Ecotypes. This is Bacillus anacrosorum. They were promoted, and they're pretty much only found at low elevations. So anyway, this is something that happened once. We'd like to know, so what is it about that ecotypes that makes it only found at the lower elevations? And I'll skip this. We've also done some genomic studies of what are defense and offense gene differences and carbohydrate utilization genes and so on. Now, I want to kind of close up with two themes about how it's useful to recognize ecotype diversity within recognized species taxa. So when we look at the ecotypes within a recognized species taxon, we find this interesting diversity, that there is one group of Atrophaeus that really likes the warm, whereas everybody else likes the cool. And if we just stuck to Bacillus atrophaeus and didn't try to delve down deeper, we'd be losing some interesting diversity. And yeah, and then there's things like, it's important when you're doing a population genetics study, trying to estimate population sizes from sequence diversity, you cannot use the species taxon for this. You really have to use the ecotypes, and this is just a kind of a population genetic reason. Also, biotechnologists, my colleague Paul Jensen has at UC San Diego, that he has focused on different ecotypes within a Salinospora, species, and he's found that the different ecotypes tend to produce different products that are useful in biotechnology. Also here, you'll see that one species, Streptococcus pyogenes, can be specialized to different host tissues. And so we wouldn't want to ignore that. And finally, in choosing vaccine targets, it's useful to focus on the ecotypes rather than the whole species, because there will be a larger set of genes that is that's held by an ecotype rather than the core genes that are held by all of them. So I think that's important to recognize in species taxa. But right now, my lab has been moving into viruses, and we've joined the Global Virome project. And the Global Virome project is really so ambitious and important. So the idea of the Global Virome project, what these people are trying to do is to discover and identify and genome sequence every single virus on the planet that could possibly infect humans. So they don't have to do every virus on the planet because they're only focusing on those virus families that are known to infect humans, not all of them. And also, with regard to mammals, they can focus on just the orders of mammals that are most likely to spill over their viruses into humanity. And that would be the bats, the chiroptera, the rodents, and the artiodactyls. Those are the three mammalian groups. And then plus birds are also important. So that's what they're doing. And the idea, and I say they because what we're doing is different. So what these folks are doing is every time they discover from some chimpanzee or from some antelope someplace, they ask, is this virus a novel virus? And what they do is they look at the sequence of this virus and ask, is it close enough to some known virus that they can just say, oh, it's in the same species as that virus and throw it away? Not interesting. So what we're doing is we're trying to focus on the interesting differences that we see within a virus species. And it's just so clear to me that there is a lot of interesting ecological diversity within what we call a virus species. And we are suffering for that right now. So we move on to comparing SARS-1 and SARS-2. So SARS-1 was the original SARS virus of 2003, which had the very nice quality that it made you feel very, very sick with muscle pains, headaches, before you became contagious. And it was so easy to contain this virus. So here, the interval between symptom onset and maximum infectivity, 5 to 7 days. That means that if you felt that you were sick, you could crawl to the nearest hospital on your knees if you had to and get there before you were contagious. Whereas SARS-2 is a different story. We now have, well, this was as of yesterday, 59.6 million cases, 1.4 million deaths, and it's still raging just because it can asymptomatically transmit. So if this is an argument that we should be studying the ecological diversity within named species of viruses, I think this is so important. So it turns out that there was a group, the Coronaviridae study group, which I believe mistakenly decided to call SARS-1 and SARS-2 the same species because they're only 2.5% divergence, and they allow a threshold divergence of 6%. So too bad for them, too bad for us. And I would say that this was a deadly consequence because people believed from what they knew about SARS-1 that this was not an asymptomatically transmitted virus. Therefore, it was really until March that we decided, oh, better wear masks. Maybe if we had not assumed that SARS-1 would predict SARS-2, maybe we would have started mandating masks a lot earlier and started saving a lot of lives. So that's what we're interested in, is finding the ecological diversity within a virus species. So I just want to mention, this isn't our work. This is work by Dan Stryker, just to say that, that this is rabies virus in bats, and this is the phylogeny of bat viruses. And it turns out that within what we call the rabies virus species, there are all these lineages of bat viruses that are adapted to different species of bats. So this lineage here is adapted to the lime green species of bat. You could read down here which species is and so on. And this one here is adapted to the brown species of bat. And this one is a generalist and so on. So we're studying West Nile virus. And West Nile virus, because birds just so wonderfully migrate themselves, that this after its introduction into the New World in 1999, birds have moved this all over, and it's in South America, it's everywhere. So this is the natural life cycle of West Nile virus, that it's really mostly a bird-to-bird-to-bird transmitted species by way of mosquitoes. So this is by, in terms of the virus, it is an ecological generalist. It can live on many species of birds, and it can be vectored by many species of mosquitoes. And then every once in a while, we get the disease. We are a dead end host. We cannot transmit the virus to mosquitoes, and likewise, horses are another dead end host. So it's a bird pathogen. So what we wanted to find out, we actually used ecotype simulation to see, looking at genomic diversity, genomic sequence diversity within West Nile, are there different ecotypes, and are they associated with different families of mosquitoes? Are the ecotypes associated with different families of birds, and so on? And I'll just skip to what we found. Oh, these are my students, Kelly, Tong, and Amy. And so in this particular diagram, each ecotype is represented only once. There's actually a lot of diversity, for example, within PE15, but to make it simple, we're just showing each ecotype once. And the pie chart is showing, oh, darn it, this isn't actually the slide I wanted to show, because there are actually 5 taxa that we studied. American crows, which are black, and blue jays, which are blue. And then we also had owls. Oh, sorry. Then we had other passerine birds that are related to crows and jays. And then we had owls, and we had eagle-like birds. And it doesn't matter if you study just crows versus blue jays, or whether you look at all 5 possibilities, you find that the ecotypes are significantly different in the birds that they tend to be associated with. So, for example, this PE39 here at the bottom really likes blue jays, but this PE3 here really likes crows, and so on, and everything in between. So that's what we found. So this is indicating that there is some, there's some ecological divergence, and it's quite significant, between the various lineages within West Nile. And maybe some of these lineages, as a result of, so what do these ecological differences mean? I should just say what that means. Does it mean that some are adapted to crows and others to blue jays? Maybe it's that when a mosquito bites a crow, this particular lineage of the virus is likely to be successful, but maybe that same lineage is not successful if it bites a blue jay. But another possibility is that the crows and blue jays might hang out in different habitats that have different species of mosquitoes. So maybe it's those mosquitoes that tend to bite American crows, maybe those mosquitoes tend to be adapted to this particular virus, and so on. So it could be a bird adaptation, it could be a mosquito adaptation, it could be both. And I'll just say something we just started to do, which is not significant yet, but we're studying influenza. And this is, I should mention, in this study, we only looked at birds from New York and Connecticut because we wanted a tight geographical sample to make our results more meaningful. And also, we started looking at influenza, which is, the reservoir is wild water birds. And in this case, from Wisconsin, we studied birds that were sampled for influenza in green-winged teal versus mallards. And this is not significant, but the number of viruses is just so small. So we're just starting this study. And basically it looks like this ecotype here looks to be about half and half, whereas these ecotypes are pretty much 100% mallard. And as we ramp up the sampling size, we we might continue to find some suggestive result. And the last thing I want to mention, which gets back to a question that was asked earlier. So we could say, yeah, so ecotypes might be the real species. They might be the real ecologically homogeneous species. But what of the species taxa like E. coli? So here, I call E. coli kind of a bad boy species taxon. It's one of the worst species taxa in the sense that it's so generalized. There are so many lineages that are ecologically distinct. In this case, non-pathogenic, uropathogenic, and enterohemorrhagic. And it turns out that different strains of E. coli might share just a small number of genes. So anyway, is there something real, biologically real, about this heterogeneous group we call E. coli? Or is there something real about Bacillus subtilis and so on? And it turns out, with thanks to the work by Kostas Konstantinidis and his colleagues over the last decade and a half, he has found evidence that there is some reality to these species taxa. And I'm going to give you at the end my commentary on it. But right now, I'll just talk about his paper. So basically, this is like his early work here, that if you take just a small number of pathogenic species, you find that basically that any two random strains, if you look at their genome sequence identity, actually look at their ANI, their average nucleotide identity, which is their average identity over the whole genome. It turns out that you're either at 95% or more, or you're 83% or less. And there's like nobody in here. And he's been saying for a decade and a half that maybe there's something cohesive miraculously about these species. And these actually are species that were recognized by bacterial systematics. And it turns out what he found early on, Kostas found early on, is that in general, a species, a systematic species taxon of bacteria tends to be 95% or more ANI, average nucleotide identity. But what we found later on is that there's something real about that. Why is they only go down to 95%? And then you find almost nothing till you get like 83%. Well, that was interesting back in the early 21st century. But then he started looking at much larger sets of genomes. In this case, he's looking at like 90,000 genomes. And again, it turns out that random pairs of genomes in the world are either 95% or more, or 83% or less. There's something just amazing about this. And so this is, he didn't really suggest a reason. And then there was later work, not much later, but by Julian Banfield's lab and Matt Ulm as the lead here. And they looked at uncultivated organisms. They looked at these MAGs, which are genomes that are assembled from metagenomes. That is, they just took these random samples of metagenome and through extremely high throughput sequencing, they produced these genomes. And turns out, when you do this without any cultivation at all, you find that the pairwise divergences are either 95% or more, or 83% or less. And it doesn't matter whether you're talking about the human microbiome, or genomes from the ocean, or genomes from the soil, or whether you're talking about this ref-seq, which are actually just cultivated genome sequences. It is just amazing. So why should this be? And Gillian Banfield suggested it might have to do, well, actually I suggest, well, did we both suggest? I guess we both suggested at the same time, that it might be due to there being less recombination at 95% ANI. Well, I've been studying this for like 25 years now, and my student Jacek Majewski and I looked at what's the relationship between sequence divergence and the resistance to recombination. And it turns out that as you look at more and more divergent organisms, you do get less recombination. So maybe it is lower recombination between these between organisms that are 95% A&I. But I was kind of suspicious about this because this is a gradual change. It just keeps going steadily up to 15% divergence, which is what we had studied. But then the Banfield study actually looks at this in a different way. We looked experimentally, what's the effect of sequence divergence on recombination rates? But what they did is they looked at what over history is the actual rate of recombination. And it turns out the rate of recombination is the purple thing here. And it turns out that in all these different groups, the rate of recombination goes down to near zero by the time you get to 90% A&I. So they suggested, well, maybe it's just the ability to recombine within what we call a species that gives us some cohesion, maybe not ecological cohesion, but genetic cohesion among members of the same species. 95% or more means that you can recombine, and they tend to be held together in some sense. And I want to just suggest and sort of close out the talk, one other way that that closely related organisms of different ecotypes might stay similar as a result of recombination. And I want to explain here that I think there might be the same limited cohesion within bacterial species taxa as we see within animal species taxa. So here I go to humans. So here we have humans, and I'm using their folklorical costumes here to emphasize that this woman here is from southern Spain, and this woman here is from Scandinavia. And they are free, their populations are free to diverge adaptively, where the Scandinavian population is extremely fair skinned, very light skinned, and the southern Spain is darker skinned for adaptive reasons. But look at what can happen in humanity and any other animal species or plant species. That yes, we can maintain the adaptive differences between these populations, but at the same time, if there is a generally useful gene that starts in one population, it can move and move and move. So what we see here is this is a transfer of the lactase persistence allele. That is, if you look here, you see that in Western Europe, there are high to moderate frequencies of this particular lactase persistence gene that allows you to utilize lactose and milk your whole life. And in other parts of the world, it's near zero or zero. So what's happened here is that apparently there has been what we call adaptive introgression, where there'll be mating between Scandinavians and people from the Baltic Sea, and Baltic Sea to the French, and French to the Spanish, and Spanish to the North Africans, and so on, that we can have the passage of one adaptive allele across the entire species, ultimately, while those groups maintain their ecological differences. And I think that might be exactly what is happening within a so-called bacterial species taxon. That we know within a bacterial species taxon, there are multiple ecotypes, and it's possible that within, so this whole box here with the blue, green, and red ecotypes, this could be like one species taxon like Bacillus subtilis, and this might be PE15, this might be PE8, and so on, that if there's a generally useful adaptive mutation, it can go from one ecotype to another, with the result, in the end, that there is a homogenization of genome, of just limited range of the genome, between the various ecotypes. So what I'm saying here is that as long as the members of a species taxon can pass among themselves these adaptive genes, that can be one of the features that allows them to maintain themselves at 95% ANI and prevents them from diverging and diverging. So I think this is one way that bacterial species taxa and animal species taxa could actually be the same in the sense that they have a similar kind of cohesion. Yes, the different individual populations within a species taxon can diverge and become quite ecologically different, but at the same time, they can share their share genes that are of a general adaptive benefit. So that is kind of the end of my story, although there's so much more to be said. And so these are my students and collaborators, which I'd like to thank. I'd like to thank them from Wesleyan, my colleagues from University of Virginia, US Department of Agriculture, Montana State University, and the DSMZ in Germany. And thank you so much for your attention. I'm so, yeah, so nice of you to be staying with me all this time, and I'd be happy to answer any questions you might have.
Oh, thank you so much, Doctor, for all this time and all this interesting stuff. So, ¿alguien tiene alguna pregunta para el Dr. Cohan o algún comentario? Adelante.
Por favor, please. Professor Doctor, amazing. It was very, very entertaining. Your tone of voice, the amount of evidence that you presented. I really, really enjoyed your lecture. And I applaud you and admire the amount of work you have put in. I do have one question that I don't know how to phrase it because I'm kind of afraid it might hurt, but I hope it doesn't. And that is, have you considered that any spontaneous epigenetic phenomena could modify your theories and hypotheses, and yours and your colleagues?
Yeah, I mean, so yeah, I think what we're looking at here are kind of the non-epigenetic phenomena. I mean, it's limited to that, but I think basically what we're trying to do is to identify those sequence clusters that are ecologically distinct from one another. And I believe, I mean, I have a colleague who you might know her by name, Sonia Sultan, who's been very famous in the phenotypic plasticity world. And yeah, every time anybody talks about something evolutionary, they're saying, but what about phenotypic plasticity? And you know, I do think about it. And I think what's probably happening is that probably as a result of changes in methylation and so on, and other epigenetic phenomena, there could be a more rapid ecological diversification than we are imagining.
Or stabilized, because, for example, I mean, as an astrobiologist, I am very, very biased towards cyanobacteria, and I find it very interesting that they are incredibly ecologically successful, even though you show that they held a hell of a lot of stability in not. I mean, they don't even try to have those horizontal transfer genes. I mean, these cyanobacteria just live happily, just switching to this one carbon molecule. That's it. I mean, they don't need to be. They don't need to try and transform themselves or provide for any genes to do any wonders, right? So I get what you're saying, and believe me, I applaud your work, and I will continue to read what you have worked on, and I just really I'm impressed. I am impressed with the amount of evidence that you have given us in this presentation. Please, Professor, you have my entire gratitude for your lecture today.
Well, Sebastian, thank you very much. That's really very kind. I do want to address one thing that you said, that from our own work in the Yellowstone hot springs, it seems that horizontal transfer is very important. And I'll just give you two examples. One is that I mentioned in passing that the cooler ecotypes, that is, as the spring has gone further and further down the hill, the cool adapted ecotypes, they have acquired a pathway by horizontal transfer for phosphonate utilization. So this was something, we just went into this so naively that we thought the main differences between the different ecotypes would be temperature adaptations. But of course, Dung, why didn't we think about that the easy mineral nutrients would be sucked up way upstream, leaving only the difficult ones. So horizontal transfer is important in that way. And I want to give one other example because it's kind of one of my favorite examples, that in both of these, so there were two major clades that I talked about in the Senecococcus. There was basically the hot clade, the A clade, and then the cool clade, the B clade, B and B prime clade. And it turns out that they are quite different in their genomes. I can't remember, maybe it's like 15% ANI, I'm sorry, 85% ANI. But what is nearly identical between them is the nitrogen fixation operon, I guess we would call it. So they both apparently fairly recently acquired this nitrogen fixation, and they are nearly identical in it. I can't remember the sequence divergence, but I think it was on the order of 1%. So it's like, you know, these are just, and I love that example because, oh no, time is ticking. If there's somebody else who wants to ask a question, I don't want to. I just want to say that this was one of my arguments for why we should understand that when there is a single gene sweep in a community, that it does not mean that there was one ecological population and there was high recombination that allowed the particular gene not to get swept. Because, you know, in this case, one of these clades is adapted to 50 degrees Celsius, another is adapted to 70 degrees Celsius. Clearly they're ecologically distinct, but there was this transfer of this adaptive mutation, which I think is very important. So anyway, thank you so much. I appreciate your questions and what you had to say. Thank you.
So I think that because of the time, I think that we should finish. So we have to let the people go. I think that there are many other questions. There are a couple of questions actually here in the chat. One related to the use of nanopore. So they are asking about the DNA sequencing technique that you used when you were working with soil. I don't know if you have any short comment to that.
Yeah, let's see. Yeah, so all of the genome sequencing that we've done has been on pure isolates. So we haven't had to deal with the complexities of a community and soil and all this. These are bacteria that we've been growing in the lab. So that turns out to be not a problem for us. And the other thing, the MinION technology works really well for pure genomes because it's so bad. It has like 10% error rate. But when you can have high throughput sequencing, it turns out you can do a majority rules and figure out with some confidence what the actual nucleotide is at a particular site. And moreover, what I really love about the MinION is that just schleps like us who don't really even know what we're doing, we just started this, we get one contig. Unbelievable. I mean, man, life is good. So the pandemic has kind of slowed us down a bit, but we're going to be adding a lot more to this. And then just one more question. So ecotypes are destined, this is from Mariana, ecotypes are destined to speciation or extinction, right? Even though they can coexist for a long time depending on use of the resources. Yeah, so I would say at the time that they're ecotypes, like one becoming two, I would call them species at that point. They're like ecological species at that point. But at some point, if they should persist, they would at some point be named different species. And if they can coexist for billions of years, they could even be different phyla. So what allows two lineages to coexist into the far distant future? And that's something that we'll just have to do next time. It's really an interesting question, but I'm glad I took that out of the talk because we would still be here till 5 o'clock.
Okay, no, and I am so glad that we will have, that we still have more topics to work on in the next meeting with you. Of course, I hope that this is not going to be the last one. Okay, I don't know if you have any final comment, Dr. Cohan, from our side. Just thanks a lot again. You are always so kind with us, always very kind and very keen to teach us and to share all this research that your team and your colleagues are also sharing with you. So thanks a lot for sharing all this stuff. They are very, very interesting.
Well, it's been my pleasure. And as I said in my introductory remarks, I would so much love to see you guys in person. How much fun that would be. Maybe we'll work that out at some point in the coming year or years.
Yeah, no, I see that vaccines are now closed. So I hope that maybe in 1 year we will have a different situation. And of course, you are always welcome here and we will be so happy having you around. Okay, thank you so much. I will let you go and thanks a lot.